All right, so we're briefly going to go over some drugs for HIV infection. We're going to look at the structure of HIV and the replication process. And then we'll go into the drug classes. So real quick, this is your basic HIV cell. Consists of a nucleic acid surrounded by core proteins, which in turn are surrounded by a capsid shell protein. That's the blue. Which in turn are surrounded by a lipid bilayer envelope. See that? The central core contains two separate but identical single strands of RNA, each with its own molecule of reverse transcriptase attached. The RNA serves as the template for DNA synthesis. The outer envelope of HIV contains glycoproteins that are needed for attachment to host cells. That's these guys right here. They consist of two subunits known as GP41 and GP120. The smaller of the two is connected firmly to GP41. So real quick, let's get into the replication cycle of HIV. So the replication cycle of HIV starts with attachment. Primary connection between GP120 on the HIV envelope and a CD4 protein on the host cell occurs. So that's supposed to be this guy right here. This is the host cell. That's CD4, a receptor on the host cell. It looks like crap on here, but it looks better in the book. So the second step is fusion. So the lipid bilayer envelope of HIV fuses with the lipid bilayer of the host cell membrane. One drug, Maraviroc, blocks HIV entry by binding to CCR5. That's CCR5, or it could be CCR4. The next step here is reverse transcriptase. So HIV is transcribed into single-stranded DNA by HIV. Reverse transcriptase converts the single strand of HIV DNA into double-stranded HIV DNA. So it goes from one strand to two strands. Fifth step here is integration. So double-stranded HIV DNA becomes integrated into the host cell DNA under the direction of a viral enzyme known as integrase. The integrase strand transfer inhibitors inhibit this enzyme. So in step 6, oh man, that looks like crap. In step 6, transcription. Some of the resulting RNA becomes the genome for daughter HIV virions. The rest of the RNA is messenger RNA that codes for HIV proteins. So in step 7, which is over here, translation, RNA is translated into HIV glycoproteins and HIV enzymes and structural proteins. So these are your glycoproteins, these are enzymes and structural proteins. So the components of HIV migrate to the cell surface and assemble into a new virus. Before assembly, HIV glycoproteins become incorporated into the host cell membrane. And that's 8A there. In steps 8B and 8C, the other components of the virus migrate to the cell surface where they undergo assembly into the new virus. So they go in here and it's breaking off. And the next step here is budding off. So the newly formed virus buds off from the host cell. And the final step here is step 10. So in this step, which occurs either during or immediately after budding off, HIV undergoes final maturation under the influence of protease. And these are the drug classes here that pertain to HIV infection. So we have nucleotide transcriptase inhibitors. The drug for that is abacavir. Non-nucleoside reverse transcriptase inhibitors. I'm just gonna let you guys pronounce it. Protease inhibitors. There's that. Integrase strand inhibitors. Yeah, I'm not even gonna try that one. HIV fusion inhibitors. And this is the one we mentioned earlier, the CCR5 antagonist. I guess I'll say Maraviroc. So, yeah, there it is. Good luck.